Have you ever looked at a cat, really looked at it, and thought, who are you? Who are you really? Well, perhaps not, but the evolutionary history of our furry friends is a very interesting story. And to do so, we will discuss primary science from the fields of phylogenetics, molecular biology, and evolutionary biology, and discuss recent advances in how new methods are used to interrogate the DNA of various extant species of felids, and how this gives us a confident picture of the evolutionary past of the cat. Let's begin by examining the Felidae family. The Felidae family arose in the Oligocene. Here we show Proailurus lemanensis, which thrived across the world around this time, with fossil evidence found in North America, Mongolia, and Europe, indicating that members of the Proailurus genus survived in a number of niches, just as today's cats do. This was the oldest known common ancestor for all extant species today, including pumas, domesticated cats, and jaguars. Much of this evolutionary tree comes from craniofacial fossil evidence, circumstantial knowledge, and some degree of hypothesis, and it's a brilliant guide to the evolutionary past of the Felidae family. Our interest begins with the species of the Proailurus genus, which lived between 30 and 20 million years ago. From Proailurus, species of the Styriofelis genus branched off within the Miocene around 20 million years ago. A less well-decorated fossil record exists, and they were thought to have lasted until roughly 10 million years ago. Our interest lies further on as members of the Felinae subfamily branched off around 12 million years ago. Genetic evidence shows 8 lineages branching off from the Felinae subfamily. The reasoning behind these 8 lineages comes from Johnson et al., who presented a phylogenetic study using genomic data from 37 living felids and 7 outgroups representing feliform carnivores. This study utilized autosomal DNA, which is DNA inherited biparentally from both lineages, Y-linked DNA, which is inherited from the male lineage, X-linked DNA, which is inherited from the female lineage, and mitochondrial DNA, which is also only inherited from the female lineage. A more recent study with a greater access to DNA samples and more advanced phylogenetic techniques from Lee et al. Agreed with these lineages, and we will show these findings as their data and dates are more likely to be accurate. The most powerful tool used in the study was SNP arrays. These are used to detect single nucleotide polymorphisms, or SNPs, at specific locations across genomes. But how does this work? Each position across the genome will consist of one of 4 bases, an A, T, G, or C. We hypothesize more closely related felid species are more likely to share identical nucleotides. For example, lions and tigers likely share many identical SNPs due to their recent common ancestry, indicating a closer evolutionary relationship. In contrast, a lion and a domesticated cat would be more distantly related and would share fewer SNPs and have a greater genetic divergence. And in this study, 63,000 unique SNPs were contrasted between 100 extant felids to assign similarities and accuracies to place them onto a phylogenetic tree. A quick sidebar here, we'll take a moment to expand on some definitions used in describing a phylogenetic tree. I copied this image online, and to use it we'll supplement species for sample. First, we have the last common ancestor for all samples, A to F. We then see a branch point created for sample B, and the remaining members of this tree are considered within the stem lineage, which will contain branch points for all other samples excluding B. In this figure, we see a phylogenetic tree, which has also, very usefully, been color-coded by continent in which the individual felids were naturally located. And within the context of this specific study, several other criteria were added to this figure. However, we are mainly concerned with the dating and lineage branch points this figure captured. The Panthera lineage branched off the last common ancestor of all extant felids roughly 11 million years ago. This includes big cats like tigers and jaguars. Then around 10.5 million years ago, the caracal lineage branched off the stem lineage. As the name suggests, this includes the caracal and other cats such as the serval. Moving on, around 9.7 million years ago, the ocelot lineage branched off the lineage stem. This includes felids such as the ocelot and the morbay. Then the common ancestor of the lynx and bay cat lineages branched off the stem lineage around 9 million years ago. And sticking to these two lineages for a bit longer, we see they did not separate until 7.5 million years ago. Going back to the remaining felids, we see the puma lineage branching off the lineage stem around 8 million years ago. This includes species such as pumas and cheetahs. Finally, we come to the final branching of lineages. 7.5 million years ago, the lineages of the domesticated cats and leopard cats parted. The latter contained species such as the Asian leopard cat. The former, named the domesticated cat lineage, contained both modern domesticated cats and extant wild cat species such as the sand cat. This is a classic example of scientific nomenclature making little sense. We have covered over 10 million years of felid evolution in the last part, and we are getting closer to the cats we share our homes with. Now let's look into the domesticated cat lineage in more detail. The jungle cat species branched off from the last common ancestor of the domesticated cat lineage 4 million years ago. Then the black-footed cat species branched off around 3.5 million years ago. And then the sand cat species branched off around 2.7 million years ago. We are inching closer to getting to our kitties. But first we must discuss something very interesting. A split occurred about 1.7 million years ago. The last ancestor of the African and Chinese wildcat and the ancestor of the European and domesticated wildcat both split into two separate branches. And these branches split again in the last million years to give rise to the individual species mentioned. However, mitochondrial DNA places Chinese and the modern domesticated cat closer, while also placing the African and European wildcat species closer. I myself would trust the information from the DNA or SNP arrays with more certainty. The authors of this study stress two caveats with the use of mitochondrial DNA for this kind of study. These two issues were A. The inclusion of NUMTs or nuclear mitochondrial DNA within the mitochondrial DNA samples, and B. The fact that felid males tend to disperse further than felid females. To briefly introduce the concept of NUMTs, as they are quite a complicated entity, over time nuclear DNA has been infested with mitochondrial DNA segments. These are largely non-functional pieces of DNA and are an area of interest within the mitochondrial research community. And within the context of the studies we discuss here, they add a level of contamination into the mitochondrial DNA samples, leading us to have doubt on the result. And to touch on the second doubt, we know that female felids tend to stay within geographic boundaries, while males tend to wander further afield. This generally makes X-linked or mitochondrial DNA less reflective of the species boundaries. So overall, the results, dates and branch points from the autosomal DNA have fewer doubts. The final question we will endeavour to address in this video essay is, how do we know that our cats come from the Near East? And using molecular biology, can we get some solid answers? To answer this, we look towards Nilsson et al., which boasted the use of over 1,000 feline DNA samples to assess their recent ancestry. We know of much archaeological evidence which points to the Near East being the genetic origin of truly domesticated cats. For example, a study by Vin et al. Found a cat buried with a human which dated back to around 9,500 years ago. There's also a plethora of archaeological and cultural evidence for cats being domesticated in ancient Egypt, which I don't think I can do justice in this genetics video. Some of the oldest findings in Egypt date back to over 6,000 years ago. Moving back to molecular biology, we should introduce short tandem repeats, or STRs. This is a useful tool in population analysis within the same species. Here, known regions of short repeats are genotyped, and individuals of the same population will have some level of variation within these STRs. This is a useful way in measuring the genetic distance between groups within a species. The study we discuss had samples from domesticated cats, wild African cats, and European domesticated hybrids. The domesticated cats were, by and large, the most heavily researched of the three species. From various sources, the authors amassed 1,018 individual cats with STR samples, 839 individuals with STR and SNP arrays, and 103 individual cats with SNP arrays. For the African wildcats, 4 individuals were SNP and STR genotyped. And for the European wildcat domesticated hybrids, 10 individuals were SNP arrayed. This was truly a large study which collected samples from 85 different sites and from over 40 countries. Based on this evidence, the authors concluded that the domestication of cats began in the Near East, which included the following countries. I hope you enjoyed this video essay on who our furry friends are. They are the descendants of an incredible evolutionary journey, which we can establish with confidence thanks to evidence from molecular biology, phylogenetics, and evolutionary biology. Please like and subscribe to support these kinds of scientific reviews. I hope to see you all next week for another journal club.